Welcome, everybody. We're going to give just a minute while people connect. We'll wait about another half a minute. We see some participants streaming in. Welcome, everyone. All right, I think we'll get started. Hello, everybody, and welcome to today's edition of Conversations on Ocean Carbon, a U.S. West Coast and Alaska perspective. This is a webinar series that aims to deliver the best available information on marine carbon dioxide removal, MCDR, and to explore concepts related to coastal ocean carbon. We're working together to create a dialogue among industry members, tribes, natural resource managers, and scientists throughout the West Coast and Alaska to provide a better understanding of marine carbon removal technologies, the state of the science, and ways to become engaged in this field. We want to thank all of you who joined us for previous webinars. Those recordings can be found on YouTube, and we will put that link in the chat. My name is Darcy Dugan. I'm with the Alaska Ocean Observing System and the director of the Alaska Ocean Acidification Network. And our network is one of 6 ocean acidification networks around the US that bring together scientists, resource managers, industry, tribes, and coastal communities to try to expand the understanding of ocean acidification and also explore adaptation and mitigation options. Change the slide here. The webinar series is co-hosted by the Alaska Ocean Acidification Network, the California Current Acidification Network, and the California Ocean Science Trust. And today I'm joined by my co-hosts, Lauren Linsmeyer and Kevin Travis with the California Ocean Science Trust. Kevin will be moderating Q&A today. And we also have Emma Stone with the California Ocean Science Trust, who will be working behind the scenes to make sure that our webinar is running smoothly. And we also want to thank Alexandra Puritz from NOAA's Ocean Acidification Program and Alex Harper with the California Current Ocean Acidification Network for supporting this series this year. So today, the title of today's session is A Closer Look at Ocean Iron Fertilization. And we have 3 speakers, Sarah Smith from Moss Landing Marine Labs, Paul McElhany from NOAA's Northwest Fisheries Science Center, and Chinmay Subin from the Pacific Northwest National Laboratory and University of Washington. We'll have 90 minutes for today's session. Each presentation will be about 15 to 20 minutes, and then we'll do a moderated question and answer session at the end. Attendees are going to be in listen-only mode today, so you can type questions into the Q&A function at the bottom of your Zoom screen, and then we'll pose them to the speakers after the presentations. We're also going to be doing a few poll questions so that we can better understand the audience here and gather some information to help put together future webinars. And we are recording this session, and we will share it on YouTube along with the others. So to kick things off, we're going to do a set of 3 poll questions. The first one is to get a sense of the audience's relationship to MCDR. So we can activate that poll question number 1. What best describes your relationship with MCDR? And we'll give everyone a few seconds to answer that. And Lauren, when it looks like you have the critical mass of answers, we can look at the results of poll question 1. Okay, so it looks like most people on the call are not currently involved in CDR, but want to track the issue. And there's also a number of people that are involved in some type of MCDR project, as well as on the regulatory management or policy side. Thank you, everybody. We have a second poll question, which is, where do you live or work? This helps us get an understanding of geographic representation and interest. And okay, looks like a lot of California, good representation from Alaska, Washington, and some from outside the United States also. Great. And our third poll question before we start with our speakers is, what is your current overall impression of ocean iron fertilization as a marine carbon dioxide removal strategy? Okay, and results there, it looks like more people are skeptical than any other feeling at this moment. And we will ask this question again at the end to see if people's impressions have changed. I'd now like to introduce our first speaker, and Sarah, feel free to turn your camera on now. Sarah is a biological oceanographer at Moss Landing National Laboratory. She studies the ecology, evolution, and physiology of marine phytoplankton and is on the steering committee of XIOS, Exploring Ocean Iron Solutions, to evaluate global change mitigation strategies, including ocean iron fertilization. And Sarah, I'll pass it to you for your presentation.
Excellent. Thank you, Darcy, for the introduction. And I'm really happy to be here today to be able to share a bit of information about ocean iron fertilization and some of the work that the group that I'm involved in, that Darcy mentioned, Exploring Ocean Iron Solutions, has been doing over the past couple years in this space. But I'll start out with just a view of the Monterey Bay. And this is actually the view of the Monterey Bay as you're coming in to port, to Moss Landing, which is where I work, kind of in the center of state of California, and those iconic power plant smokestacks, which if you've ever been out on the Monterey Bay on a rough day, are a very, very welcome sight. And this is taken from the bow of our small research boat, named the RV John Martin. So for those of you who are familiar already with ocean iron and John Martin's role in that, this vessel is named after him. Okay, so I'll dive right into it, first by introducing our group, EXOIS, Exploring Ocean Iron Solutions, because this group has been pretty active in having conversations around charting a path forward to explore kind of a responsible path to obtain the information needed to assess whether ocean iron fertilization is useful or valuable. So I'll be referencing some of these documents throughout my talk, but I wanted to upfront provide the link. Our website has all these resources that are downloadable. First, the Ocean Iron Fertilization White Paper, which was a document that we created in fall of 2022, that kind of outlined our plans at the time. We held a workshop in Moss Landing with about 35 to 40 oceanographers, social scientists, policymakers. And from that workshop, we published a report called the Paths Forward document. And this is a much more detailed version of a paper that was just released last week, or September 9th, in Frontiers. So if you're looking for more information than I'm able to share in this short presentation, please do check out those resources. We also held a small field planning workshop in May of 2022, with this group pictured here, to start to detail what an actual at-scale field experiment and what the next generation of field experiments should look like. I'll talk more about that as we move through. But I'll start here with kind of the things that I'm going to cover. I'm going to talk generally about why marine carbon dioxide removal is needed, what ocean iron fertilization is, what some of the applications could be, the potential for carbon drawdown, what sort of previous field trials have been done, and what projects are underway, and where ocean iron fertilization could be applied in the oceans today. Okay, so first, why carbon dioxide removal? Well, this is probably not a big surprise to the people that are in this audience, but CO2 levels in the atmosphere are increasing at a relatively unchecked pace and causing all sorts of problems for global warming, stratification, ocean acidification, and we have concerns about sea level rise, biodiversity loss, intense drought, fires, and all the problems that are accompanying that. So the time to decarbonize and to stop relying on fossil fuels has come and gone, and we have an additional problem. Even if we ceased to burn fossil fuels to meet our energy needs, the projections are that by including all sorts of non-fossil fuel energy or sustainable sources to kind of wean us off fossil fuels, we still need a way of removing the excess carbon dioxide in the atmosphere if we're going to achieve net zero in 2050, which is the goal to limit warming to 1.5 or 2 degrees C above pre-industrial levels. So carbon dioxide removal, whether we're talking about terrestrial or marine forms, is now necessary. And the ocean has a particularly important role in in the globe for a potential to store some of this excess carbon. So this is a graphic that's showing where most of the carbon on Earth can be found. And you'll see a relatively small amount is in the atmosphere. The small amount that we have added relative to the total global amount of carbon is causing problems. But the deep ocean has roughly 38,000, 37,000 gigatons of total carbon. So there's a lot more potential to store the small amount of damaging carbon that's present in the atmosphere in the deep ocean. That's basically the reason why marine carbon dioxide removal is so attractive. The National Academy's report that was released in 2021 in December identified 6 kind of main technologies or approaches to store carbon in the ocean. And one of them is nutrient fertilization or iron fertilization, which is what I'll focus on today. And the premise is essentially the addition of trace amounts of a limiting nutrient, the micronutrient iron, in certain regions of the ocean that stimulate phytoplankton productivity. Phytoplankton then, through photosynthesis, sequester carbon, and if they die and sink into the ocean's interior, that carbon is removed from the atmosphere. It's one of a suite of several other MCDR approaches. So what is ocean iron fertilization and why is it something that we could consider in this context? Very quickly, all cellular life relies on iron. It's necessary for respiration and photosynthesis and nitrogen fixation. The enzymatic reactions that catalyze it rely on having access to that trace metal. And cellular life evolved in a really high iron and relatively low oxygen world. So our cellular demands for iron are pretty essential. As photosynthesis evolved and the world became oxygenated, the cellular machinery the iron oxygen changed the solubility of iron in seawater and reduced the ability of life living in the surface ocean to obtain it. So what you see in the geological record are these banded iron formations where iron oxides were precipitating out of the ancient ocean as life was becoming accustomed to the high oxygen levels. Consequently, much of the surface ocean is limited for iron. Iron's not something that we think of being limiting in terrestrial ecosystems at all. Iron's, I think, the third most abundant element in the Earth's crust. But the farther you are away from the Earth's crust in the open ocean, iron can actually limit all of cellular life. And that's what's being shown here are inputs of iron to the open ocean. So innovations in analytical techniques and the ability to sample some of these difficult environments pioneered at Moss Landing Marine Labs and pioneered by John Martin and his team allowed us to develop the first vertical profiles of iron availability in these regions. And that's what you're seeing here. This is a depth profile of iron concentrations in the water column at Ocean Station Papa, which is in the subarctic Northeast Pacific. And iron, like these other nutrients, follows a nutrient-like distribution, meaning that it's scavenged by biology in the upper ocean and increases in concentrations with depth. What was interesting to note is that iron levels were super low and there was plenty of available nitrogen or nitrate all the other fertilizer that phytoplankton need. There are lots of regions in the ocean that are like this. This map over here on the right-hand side is showing that. You see all of the warmer colors are where there is a lot of nitrate. And these are not the amount of nitrate that can be measured in these regions doesn't support chlorophyll or phytoplankton biomass at the level that you would predict. So these regions are identified and kind of defined as HNLC, or high nutrient, high nitrate, low chlorophyll regions. These regions are where phytoplankton productivity is limited because of reduced iron supply. So with all this in mind, and all this data kind of in hand, John Martin put forward this iron hypothesis in 1990. It was very famously attributed to this quote, "With half a shipload of iron, I could give you an ice age." Because John Martin recognized that iron supply to the surface ocean over glacial and interglacial scales was a mechanism by which the ocean actually could regulate global climate. So that when CO2 accumulates in the atmosphere and Earth dries out and it gets warm, there is an input of iron into the surface ocean, which can stimulate phytoplankton production. Phytoplankton then sequester carbon, sink to the deep, cool things back down. And this is a mechanism by which Earth could actually regulate global climate. So he developed this iron hypothesis, which was that iron deficiency limits phytoplankton growth, the uptake of CO2, and the flux of carbon. Iron limitations responsible for the persistence of these HNLC zones found over in 30% of the oceans worldwide. And natural iron supply during glacial maxima contributes to the drawdown of atmospheric CO2. The corollary of all of this is that large-scale purposeful iron enrichment could help control atmospheric CO2 if we do it right, if we do it the way that the Earth knows how to do it already, by sequestering carbon in the deep waters and the bathypelagic sediments. But that corollary is not part of the original iron hypothesis. Okay, so this graphic is taken from the Ocean Visions, and it shows basically the principle where if we're going to actually use this ocean iron fertilization as a tool to remove CO2 from the atmosphere, you would fertilize certain regions of the ocean, either with a ship or maybe a plane or some other kind of mechanism to seed the surface ocean, stimulate a phytoplankton bloom, and then that phytoplankton bloom sinks or is processed away. The farther it gets into the ocean's interior, the longer it's sort of locked away from the atmosphere. So the more durable that carbon sequestration could be. That's the principle. So what kind of carbon drawdown could ocean iron fertilization achieve if it were actually used in this kind of sense? Well, first of all, let's define the problem. We need to take tens of gigatons of carbon out of the atmosphere per year in the coming decades. And really, no single carbon dioxide removal approach, including the terrestrial ones, is likely to reach that capacity. For ocean iron fertilization, all we have are small scale field studies and kind of estimates of carbon export from different regions of the ocean to evaluate its efficacy. But this has never been done at scale. And so we need to rely on models that will help constrain some of these kind of estimates of how much carbon can be removed. A couple of references here I've included. This is an older one, but it's a very nice ocean model that suggests that to completely eliminate iron limitation, I think in the global oceans, I can't remember if this is Southern Ocean or global oceans, it only gets us to about 1 petagram or 1 gigaton carbon per year. Carbon export across the 100-meter water depth, so that's more durable carbon sequestration, gives us about 2 gigatons or petagrams per year in a more recent modeling study. So that's not anywhere near the tens of gigatons of carbon that need to be removed. But, you know, this is sort of a situation where it's not going to be any single CDR approach, most likely, but a suite of CDR approaches combined, obviously, with the very first step of decarbonization that may be needed to get us to where we need to be if we're still aiming to achieve net zero by 2050. The interpretations of oh, sorry, backwards, of if this is enough for this technique to be considered are a little bit in the eye of the beholder. And that's why I've put this graphic here. Do you see two faces looking at one another, or do you see a vase? Kind of depends on your perspective. And here are a couple quotes where, people have said that iron fertilization can't reduce atmospheric CO2 to significantly alter the course of climate change. Maybe it can't if we don't decarbonize, but at this situation that we're in, maybe it's necessary to slow down biodiversity loss and climate change. Okay, so here is a summary of where kind of field trials for ocean iron fertilization have been conducted. And I want to take a moment to be clear that no field trials of ocean iron fertilization for marine carbon dioxide removal have been conducted, but these are all field trials that have been conducted to address John Martin's iron hypothesis, which is most simply that iron supply governs phytoplankton primary productivity. And I don't think that there's an oceanographer alive who would contest the results of all of these studies, both artificial ocean iron fertilization and natural, looking at the inputs of iron from other natural sources. It is inconclusively true, or conclusively true, that iron supplied to the surface ocean controls phytoplankton productivity. What is less certain is how much of that phytoplankton productivity is exported, how much of that carbon is durably sequestered, and kind of even more uncertain is, can we as humans engineer a bloom that would be efficient to sequester carbon at the scales that we need it to. So, to summarize some of the projects that I'm aware of that are underway, and really I'll lean mostly on what I've been involved with EXOIS, but I wanted to mention a couple others. There's a Korean iron fertilization experiment um, in the planning stages, KIFES, and there's a really nice paper, Yoon et al. 2018, that summarizes all of these field experiments as well, that describes that effort a little bit more. I know about some of the work of Sir David King, who's the founder and former chair at the Center for Climate Repair in Cambridge, who's been interested and involved in this space. And EXOIS, which is the group that I'm involved in, I'll summarize a bit of our activities in a minute or two that I have left. So EXOIS is an umbrella organization. It's not a funding agency and it's not a formal group. But we have a bunch of different individuals who are members that work on different aspects of ocean iron fertilization. We meet monthly online for forums that cover a variety of different topics. As I alluded to, we had a couple planning workshops. This is all in kind of the early stages. We have recently started monthly meetings with social scientists and governance experts to start to really flesh out strategies to include those very important aspects into some of the field planning that we think will be necessary to evaluate this technology. And then there's also some natural science work that's happening, investigations in the different forms of iron. We call that like the Bake Off, like the British Bake Off. What form of iron might work? We've used iron sulfate in the past. Is that the best? Can we engineer a bigger batter bloom with something else? A lot of that work is being done in cultures or with mesocosms. And then really the bulk of the work, I think, to date on this effort has been in the modeling space, the global Earth atmosphere models and kind of these regional Aussie type models. The workshop activities have been largely funded by Astera, which is a kind of science incubator. Just to be transparent about the funding, I believe that we have more information about who has been funding this kind of workshop and planning stage effort work. But to be clear, the field work, none of that is funded. So none of that is moving ahead in any kind of real way at this time. So our plan, I'll summarize just really briefly. We think that in order to evaluate ocean iron fertilization, that a new generation of larger kilometer scale and longer field studies need to be conducted to address the remaining knowledge gaps and uncertainties. And this is basically to address the MCDR aspect that the original ocean iron fertilization experiments were never designed to assess. Towards that, we've made some progress out in down selecting of these HNLC regions where we might target. So the HNLC regions are largely the subarctic Northeast Pacific, the equatorial Pacific, Southern Ocean, and then there's what we call LNLC or low nutrient, low chlorophyll regions, like the middle of the oligotrophic gyres, which are also limited for iron. We've chosen to move forward with a idea to fertilize a patch in the subarctic Northeast Pacific, and that was driven by a lot of logistical and practical and kind of ecological constraints. More information can be found in the reports, and I'm happy to chat more about it. We're going to focus on using iron 2 sulfate, perhaps with sulfur hexafluoride as a tracer, because we know that that works. We're going to do a much larger patch size relative to what's been done before. We're developing a suite of measurements that we call MRV for carbon, or monitoring or measurement, reporting, and verification to assess how much carbon sequestered. And then, very importantly, a suite of measurements called EMRV, or ecological and environmental monitoring, reporting, and verification. We think that that's as important as MRV is for carbon accounting because of the potential to impact these ecosystems in ways that we might perceive as negative. So we're in the process of doing an EMRV kind of co-design with social scientists, communicators, and stakeholders, key stakeholders. And I'll just wrap up with the perspective that we're doing this all within a set of guiding principles that can also be found on our website, which is to prioritize collective benefit for humans and the environment, establish clear lines of responsibility, and commit to open and cooperative research, engage the public, and evaluate what we are doing iteratively. This is not a commercial endeavor, and we are seeking funding that's aligned with that in mind as well. So that's my last slide. I'll acknowledge all my co-authors and funding agencies once again, and I'll stop there. Thank you.
Thank you, Sarah. Thank you for that presentation and for setting the stage here for us. We'll be taking questions in the question box. We see a number of questions have come in, so you can continue to add your questions and we'll bring them up during the question and answer session. Our next speaker is Paul McElhany, who will be talking about risks to the ecosystem from ocean iron fertilization. Paul is a research ecologist at the NOAA Northwest Fisheries Science Center in Seattle, where he started over 25 years ago, working on evaluating extinction risk and recovery planning for Pacific salmon. His research focus for the last 15 years has been on understanding the ecological effects of ocean acidification through experiments and modeling, and now has expanded to include research on the ecological risks and benefits of marine carbon dioxide removal. So I'll hand it to you, Paul.
Thanks. Get my slides up here. Can you see those?
Yes, looks great.
Great. Okay. Yeah. So Sarah did a great job of kind of laying out what iron fertilization is and how it might work. I'm going to focus on particular potential ecological effects of iron fertilization. But I want to start with this slide, which is a variation on a slide that Sarah showed, to kind of emphasize why it is that we're looking at iron fertilization. As I said, I'm going to sort of focus on ecological impacts, most of them risks from iron fertilization. But I want to kind of highlight that there's a risks of not removing CO2 from the atmosphere, which is increased climate change and acidification. This slide, as Sarah also kind of illustrated, there are a number of different methods, marine carbon dioxide removal techniques. This slide is kind of built off of some graphics and a framework of putting the methods relative to the cost in terms of kind of cost per ton of carbon removed, and effectiveness in terms of their ability to sequester CO2. That's in the NOAA strategy for carbon dioxide removal research. So the methods include ocean alkalinity enhancement, and there have been other presentations in this series about these different methods. Electrochemical methods, which may be fairly effective, but costly. Artificial upwelling and downwelling. Iron fertilization, lower relatively on the sort of cost axis, kind of middling on the effectiveness. But again, there are large error bars around both of these costs and effectiveness sort of assessments. And macroalgae cultivation, so rearing seaweed and then sequestering it, often by sort of sinking it in the deep ocean. I also want to kind of call out coastal blue carbon, which NOAA is considering separately from the MCDR techniques, and that the blue carbon, once those kind of habitats are established, they should be able to continue sequestering carbon without continuing human intervention, unlike the other CDR methods. So each of these methods, one thing I think to sort of emphasize about all of these MCDR methods is the scale at which they need to be applied to achieve the amount of carbon dioxide removal that's necessary to change sort of our climate change trajectory. So the models project that we need around 10 gigatons of CO2 sequestered by 2050 per year. These are just some kind of extrapolations based on the scale of effort that would be needed to remove about a gigaton per year using any single method. As Sarah mentioned, no single method would likely be or could be kind of applied to remove all the CO2 that's necessary. So most sort of scenarios lay out a mix of different methods. So alkalinity enhancement would require a large number of ships, a big increase in lime if that's the material that would be dispersed. It's one of potential. The electrochemical methods require a great deal of electricity to pull them off. The macroalgae approach would require a large area of kind of the ocean converted to production of macroalgae to sequester at 1 gigaton of scale. As Sarah mentioned, iron fertilization would also need to occur at a large scale, essentially removing iron limitation in the kind of world's ocean. And from that, you might get kind of a gigaton or two of CO2 removed per year. It would have a sort of a ceiling in that range. Each of these methods has ecological risks above and beyond sort of what they would do in terms of changing the atmospheric CO2 concentration. In this table, which is modified from a table that we put together in the NOAA CDR strategy, has each of the rows is one of the methods for MCDR, and the columns are potential ecological effects from those methods. As you'll see, iron fertilization has a lot of X's. There are a lot of potential ways that iron fertilization could impact the environment. One thing that we added in this version of the table is the effect of not doing MCDR, of not reducing, using some CDR method to reduce our CO2, which is continued climate change and ocean acidification. Okay, so in the next set of slides, I'm going to sort of list through some of those X's that were in the previous table about ways that iron fertilization could affect the environment. And one of the kind of key issues of concern is called nutrient robbing. So as the phytoplankton abundances are increased by reducing iron limitations in the places where iron is currently limited, sort of illustrated here in this bottom left kind of map of the world. In the red areas, it will consume other nutrients, nitrogen and phosphorus, that in the absence of the iron fertilization would naturally be transported to other parts of the ocean where those nutrients would provide sort of food for phytoplankton that are growing there. So this is a map of a model output that simulated iron fertilization on top of climate change. And the red areas, kind of in the bottom left, show areas where net primary productivity, phytoplankton production, would be increased. And the blue areas show areas where it would be decreased because of the decrease in availability of nutrients in those regions. One thing to note is that these regions that would be under this model would have lower primary productivity are some of our most sort of productive regions, like here along our west coast in the United States or along the west coast of South America, these highly productive kind of upwelling regions. So that's kind of one factor to consider. This particular model took a particularly sort of pessimistic scenario, and that it applies the iron fertilization on top of climate change, with the kind of modeling consequence that climate change increased stratification, and it itself was already sort of leading to nutrient supply issues in some of those coastal regions. This map here on the right, they coupled their model to one that looked at higher trophic levels, and it sort of shows under their scenarios the situations in which you'd have an increase of animal biomass in under climate change only or under iron fertilization. The sort of locations where you'd have kind of winners in a climate change and iron fertilization scenario are limited to kind of areas around a few islands. It's not a very optimistic perspective. Okay. Another kind of topic of concern with iron fertilization is increase in harmful algal blooms. So as you add iron, the goal, the purpose of iron fertilization is to increase phytoplankton productivity. Some of the phytoplankton that may be increased in abundance produced harmful chemicals. In particular, the concern is about Pseudonutia, members of the Pseudonutia genus that produce domoic acid, which is a neurotoxin that can be amplified up through the food web. Kind of here on the West Coast, where lots of scenarios, lots of situations where this harmful algal bloom and the presence of domoic acid have resulted in closures of crab fisheries and shellfish fisheries along our coast. And so concerned that this might kind of increase. Here on the right, this figure is a study that was done some years ago where they went to kind of one of these iron limited areas, took sort of phytoplankton up from the ocean and did rearing on deck, where they added different amounts of iron, and in one case iron and copper. And from this, they saw kind of an increase in the presence of domoic acid from this sample. So the extent to which this would happen at a large scale is kind of is unclear, but it's an area of concern. So as the phytoplankton take up CO2, they sink into the ocean, and as they sink, the phytoplankton and the zooplankton productivity that's prompted by that iron addition will be decomposed by microbes, and in that process, they consume oxygen and release CO2. So in some areas, at some level of the ocean, there would be a kind of a decrease in the amount of oxygen available. That's sort of shown if we look at the graph here on the right. This is showing some sort of modeled effects of changes in oxygen concentration. In the upper graph, the dashed lines are the decrease in the Southern Ocean, and the solid lines are global. And the colored lines are sort of different numbers of years of which iron fertilization is occurs. In this sort of broad scale global modeling, there is a decrease in oxygen, but at that sort of global average scale, you don't kind of get near the thresholds for hypoxic events, except the deep ocean. The bottom graph here shows sort of suboxic volume. That's the kind of volume of the ocean where the oxygen concentration gets very low, less than 5 nanomillimoles per cubic meter. And so this would be kind of in the deep ocean. You'd have an expansion of that low, very low, almost anoxic volume of water as that sort of biomass sunk to the ocean. Okay. Sort of likewise, you'll have sort of changes in pH associated with the blooms and then the sinking of those blooms. So what the CO2 iron fertilization does is increases the primary productivity, and that those phytoplankton will through photosynthesis pull in CO2, which will increase the pH at the surface. As that material sinks, again, it will sort of absorb oxygen and release CO2. So you can have an increase in acidification at some depths, sort of a few hundred meters down, most likely, as sort of illustrated in this modeling output here on the, this has got sort of depth on the y-axis and then latitude on the x-axis. And in the Southern Ocean, which would be sort of most affected, you have this decrease in the pH, so increased acidification at this level where the phytoplankton are decomposing. This model sort of shows an overall increase in the pH at the higher, at the lower latitudes from the drawdown in CO2 from the atmosphere. This particular question about how these marine carbon dioxide methods will affect ecology in the region is the focus of some of our research here at the Northwest Fisheries Science Center. We're focusing on the situation where you have high pH and high alkalinity conditions. These sorts of scenarios are most common with the alkalinity enhancement and the electrochemical methods. But with iron fertilization, you'll also have this sort of shifts in the pH of the environment at a smaller local scale, where during the bloom you could get significant drawdown in CO2 and increases in pH, and then as that sinks, you could get significant decreases in pH. Okay. Another kind of potential effect of the iron fertilization is production of other greenhouse gases. So sort of a byproduct of the phytoplankton chemistry and its interaction with microbes is the production of other greenhouse gases like nitrous oxide or methane. And that's sort of illustrated here in this graph on the right, which shows a result from an experiment where they did iron fertilization, which sort of is shown by the arrows that are at the top of the figure over time. And as expected, you see the increase in chlorophyll over time, which are the open square dots. But you also see this increase in the nitrous oxide production. Nitrous oxide is a kind of more like 300 times as effective at CO2 at trapping greenhouse gases. So while it might be a small amount, it sort of reduces the effectiveness at sequestering CO2. Maybe on the order of sort of 10% of the amount of CO2 is sort of offset by this production of nitrous oxide. Other compounds that are produced as a kind of byproduct of phytoplankton metabolism are MDS. MDS is a compound that is not a direct kind of greenhouse gas itself, but the sulfur compounds, when put into the atmosphere, can lead to changes and increases in sort of cloud formation. And so it's thought that the increases in sulfur from MDS may affect the cloud cover, which could reduce the amount of heating kind of on the Earth. Though this particular model and the example that I've shown, they found that there was a reduction in DMS production from iron fertilization in the Southern Ocean, which would lead to less cloud cover potentially, and therefore more heating. Okay. One kind of question that has not been as much of an issue in the small pilot studies that have happened so far, they've used relatively clean sources of iron. But if you were to do this at scale, there's a concern about whether or not the iron material that you were dispersing would be sort of completely clean, or whether or not it might have contaminants like heavy metals or copper, other things that could affect the ecology. This has been a concern for electroalkalinity, for alkalinity enhancement methods, such as dispersing olivine, where it's concerned you might put nickel or chromium. I've seen references to this in the kind of literature on iron fertilization and in discussing it with other scientists, but I haven't seen a good review of this issue of what sort of the sources of iron would be and how we can sort of ensure that they would be free of impurities. Okay. One kind of case that's been put forward is that iron fertilization could have a fisheries benefit, sort of shown by this kind of simple diagram here on the left. Ocean seeding, which is how they're referring to iron fertilization, it'll lead to increased phytoplankton production, which would then lead to increased zooplankton. Then fisheries populations feeding on the population phytoplankton and zooplankton would increase in abundance. This is a graphic that I took from Ocios, was proposing doing this off of the coast of Peru as a means of fisheries enhancement. One kind of in the upper right, showing a kind of release of iron sulfate as part of a controversial experiment that was done off Haida Gwaii in 1912. This is an experiment that wasn't done to sort of test the iron hypothesis as laid out by Sarah. This one was done kind of explicitly to try and enhance fisheries in that region. There was very little sort of scientific monitoring of what happened during that experiment, and so there's sort of no assessment of whether or not it did what it intended to do. One kind of case that is a natural kind of increase in environment, a natural increase in iron, that could be an analogy of what happens with human iron fertilization, happened with a eruption of a volcano in the Aleutian Islands, which released a lot of iron into the North Pacific. And there was some kind of argument that this iron led to an increase in sockeye production in the Fraser River, sort of following that eruption. Sort of further analysis has kind of argued that it's really hard to conclude that you did see some sort of increase in that fishery. So it's really sort of unsure exactly whether or not there would be kind of fisheries benefits associated with the iron fertilization. One thing to sort of keep in mind in trying to make these higher trophic level predictions about what would happen, I think is kind of illustrated by a case study or an example, I guess, of what happened during the 2016 heat wave here in the North Pacific. It led to a real increase in the density of pyrosomes in the region. Here you can see them kind of being pulled up on a fisherman's net. They were, kind of a problem in clogging nets. The diagram here on the right is a food web diagram of the region, where each of the circles is a species or a species group. And the size of the circle is a reference to how much it changed in response to that heat wave. The biggest thing that stands out is this increase in the pyrosomes. The key issue here is not that I think that pyrosomes would increase necessarily in the response to iron fertilization. It's just that we didn't predict this. No one and none of the models really predicted that in the event of a large-scale heat wave, you would get this huge increase in the pyrosome production. So I think it argues for a lot of humility in our attempts to predict what we think will happen at these higher trophic levels when we do large-scale perturbations. Okay. I've given the sort of rain on the parade story on iron fertilization in a lot of my previous slides. I want to kind of come back to this issue of the cost of doing nothing, which is that the projections about what happens with increased climate change and increased acidification. These are results from some models showing change in biomass with increased climate change. And you can see kind of in this bottom left scenario, a big decreases in biomass in some of our most productive fisheries areas here on the West Coast, Europe, the West Coast of South America. And this diagram on the right, kind of emphasizing again, trying to look at the comparisons between the effects of large scale extreme events and changes in the mean, both of which have kind of impacts on declining biomass around the world. So there's a cost to doing nothing. So the challenge is weighing the potential cost of doing nothing and the risks from potential iron fertilization methods. So kind of one more topic that I want to hit on before the summary. Because I said that I would do it. It's not really my area of expertise, but it touches on what we do here at NOAA. So I wanted to kind of mention it, which is the legal, regulatory, and permitting framework of ocean iron fertilization. A lot of these actions would happen in international waters. The kind of key targets for this are the Southern Ocean and the kind of Northeast Pacific, as well as sort of the center of these other gyres, which tend to be in the middle of the ocean. So there are a few laws that apply here internationally, but they're developed generally in the framework of ocean dumping, so preventing people from dumping things into the ocean which are hazardous. So there are two kind of key laws, the London Convention and the London Protocol. U.S. is party to the convention, but not the protocol. The protocol is sort of the more strict of the two. Here in this table, it shows under the convention, these are the things that are sort of explicitly prohibited. If they're not prohibited, there's kind of not a regulation to not dump them. The protocol says that you shouldn't dump anything except the things that are on this list, which are possible under the protocol. And in an amendment to the protocol explicitly looking at iron fertilization was put forward in 2022. It's not in effect because it hasn't been passed by enough countries, but it's generally sort of saying that it shouldn't be done except under authorized permit. I put a quote from that amendment there in the bottom right. It says in geoengineering activities listed in Annex 6, Annex 4, the only things listed there are iron fertilization. In U.S. waters, this is covered by the U.S. Marine Protection Research and Sanctuaries Act, which has the EPA as a lead. And NOAA is consulted under that act through our kind of mandates under the Endangered Species Act, the Magnuson-Stevenson's Fisheries Act, the Marine Mammal Protection Act, and the National Marine Sanctuaries Act. There are also tribal treaties and rights that apply to activities that happen in the U.S. water. I think a key point here is that there's not really a structure that's set up and enforced to regulate the iron fertilization on the open sea at the moment. Okay, so last slide is sort of a summary. Iron fertilization is a strategy to change the abundance and distribution of species at the base of the marine food web at a scale that affects the global carbon cycle. So it's kind of a big thing. There's substantial risks from iron fertilization through a variety of mechanisms, but there are also substantial risks from increased climate change and acidification. We need a better understanding of the risks and benefits before large-scale sustained deployment, but we won't really know the impact of these large-scale sustained deployment at like an entire sort of Southern Asian scale until they happened, because they're what happens at smaller scale sort of pilot studies may be qualitatively different from what happens at a large scale. So I'll stop there.
Great. Thank you very much, Paul. That was a lot of ground to cover, and we really appreciate it. There have been a lot of questions coming in through the question box. Please keep those questions coming. We're going to make some time at the end to make sure we can address some of those. We'd like to welcome our third and final speaker, Chinmay Subin, who will be talking about new research underway to try to make ocean iron fertilization safer. Chinmay is a research scientist at the Pacific Northwest National Laboratories and an affiliate professor in the Department of Material Science and Engineering at the University of Washington. She is interested in addressing materials challenges related to energy, water, and sustainability, and her current research is focused on marine carbon dioxide removal and critical mineral extraction from domestic sources to support U.S. national decarbonization goals. And I will hand it over to you.
Thank you, Darcy. So I think the previous two speakers did a fantastic job of explaining why we need marine carbon dioxide removal in general, in addition to emissions reductions. And also we heard about the potential for impact both on the ecosystem and also in terms of carbon removal from ocean fertilization. So my presentation is going to be a slightly different flavor, and I'm going to build off of some of the content that the previous speakers shared. So with that, for those of you that are not familiar, Pacific Northwest National Lab is one of the DOE national laboratories. We are the only DOE national lab with a marine sciences facility. So what you see there is a picture of our lab in Sequim, Washington. And the lab actually works on a variety of different marine sciences related research, in particular energy and climate solutions, equitable economic development for coastal communities, and what we see and ecosystem related, healthy ecosystem related science. So these three key core goals of PNNL SQUIM are things that anyone thinking about MCDR probably has front and center in their mind. These are things that that come up often in terms of safety and community willingness to try these interventions. So we're thinking about these even outside of MCDR broadly for coastal communities. So with that, I'm going to jump into some of our high-level research priorities. PNNL's MCDR research, we work on everything from very early stage ideas, so you could have a very new idea that needs to be tested, so proof of concept, all the way to much more mature technologies that we want to demonstrate, test the safety of. And we do all of this in engagement with our local communities and stakeholders. So questions that we ask when we go through these projects include, will this work? Is it safe? And does it fit within our existing resources and community priorities? So here are some examples, images of things that we've been working on, but this is a process. It's not a individual project. It's a lot of interdisciplinary work with scientists, technologists, and social scientists. So PNNL has many different marine carbon dioxide removal projects. Here are the various pathways in this graphic. We work on several of them. I'm not going to go into all the details, but for the purposes of this particular webinar, we're going to focus on ocean fertilization, which our previous speakers really well explained. And what we know to be true is that there is very high estimated CO2 removal potential for ocean fertilization, but there's also lots of concerns around ecosystem impacts if the approach was implemented at scale. So at PNNL, what we're trying to do is explore alternate ways of delivering the iron. And here's why. So one reason is to improve nutrient use efficiency. So if we increase the efficiency, that translates to reduced amount of nutrients added to the environment and any downstream impacts from that. So that's one reason to improve efficiency, and I'll talk more about it in a minute. And the second reason that we're thinking about alternate ways of delivering the nutrient is because we are interested in delivery methods that give us a lot more control over stopping immediately if we see any downstream impacts, as opposed to it was an experiment done and then there's nothing we can do about it, right? So can we build that control in from the start? That's kind of the question we're interested to address. So just quickly, we're interested in carbon removal. Carbon removal is tied to the amount of biomass, and the biomass is then tied to the availability of nutrients. So if we think about nutrients, we've been talking about iron, but photosynthesis is dependent on several other nutrients, which we refer to as trace nutrients, iron, zinc, manganese for key functions. And it's not just their availability, they have to be bioavailable. This could mean that they have to be in the right oxidation state. It might have to mean that they are not bound by other things in the ocean that prevent them from being taken up. So previous experiments were not necessarily done with accounting for carbon capture with iron addition to the ocean. We heard that multiple times before. What has been true, however, is these studies used large quantities of iron that were added to the ocean. They showed significant increase in phytoplankton productivity. But they weren't designed to remove CO2, so they didn't really track the efficiency there. But one of the things to notice is also there was no control over what happened to the iron, whether it drifted down to the bottom of the ocean or became unavailable. In fact, the National Academies of Sciences reports that most of it actually wasn't really available for phytoplankton growth. 75% of it just was wasted in the sense that it was lost to the local environment after the addition wasn't translated to growth. So what we're trying to do is ask the question, how could we increase the efficiency of the iron that is added to the ocean? And how do we, in parallel, minimize the ecosystem impacts, negative impacts that might come from it? So if you look at literature, here's a study where they looked at potential ways to increase iron use efficiency. As you can imagine, if you increase the lifetime of iron in seawater, so keep it in the right oxidation state, keep it bioavailable in the right part of the ocean where there's phytoplankton growth, then you can increase the productivity. So in this particular effort, they suggested that you could take nanoparticles of iron and coat it with multiple different layers to protect the iron and increase its lifetime. So you could think of this as the concept of slow release nutrients or slow release fertilizers people might use in terrestrial settings. But the problem is, in addition to the iron, this means when we scale the process up, we're also adding polymers and chelators and a whole bunch of other things along with the iron, the impacts of which may not be well established. And when we add nanoparticles, we still might face the same challenge of where do they end up and how do you track what was added. So for example, chelators, here's the example of EDTA, which is a very common chelator, is known to extend the lifetime of iron in seawater or other ions as well, in addition to other metals as well. So EDTA is very common. It's used everywhere, detergents, cleaning, and if you pick up your bottle of soap or shampoo, you will see that EDTA is a key ingredient in it. So this is everywhere. So we wanted to see how effective a chelator would be in extending the lifetime of iron. So we did a quick experiment. What you're seeing here is a phytoplankton. We used a model organism called Picochlorum cellurii, a microalgae, the photograph on the left-hand side. And then we added iron chloride, both with EDTA and just iron chloride as is. And in the middle panel, what you see is the growth. We're using a light-based technique to measure the density of the culture. How fast is it growing? And what you clearly see is that with the EDTA, the growth rate is really rapid. And in the final panel, what you're looking at is that the amount of biomass, which really translates to the amount of carbon, is correlated with the presence of EDTA. It's much higher in the presence of EDTA than without. But turns out EDTA is not great, it is not biodegradable, and it's highly persistent in the environment. It's something we really want to avoid. So we wanted to not rely on a chelator to extend the lifetime of iron, but we wanted to find something different. So can we eliminate EDTA? So we went back to the drawing board and we thought about what does ideal iron addition look like from a point of view of having the high level of control so we can stop the process or control very low dose delivery. So it turns out phytoplankton don't need a lot of micronutrients, but they need it where they can use it, which is really top surface where there's sunlight. They need it in a form that they can utilize it. So if it's reduced, they can utilize it much more readily and just when they need it. So if there's excess amounts of nutrients, it may not always be efficiently used. So we need it close to the surface, parts per billion, very low levels of iron. Needs to be bioavailable, delivered in proximity so it can be utilized more rapidly. Frequency, instead of large amounts of iron added infrequently with ship or airplanes, could we do this on a more frequent, low concentration basis so the uptake and the impact locally is not as drastic? And with the hopes of minimizing any localized impact or downstream impacts. So our goal is really to develop a highly controllable, low-dose delivery method. The approach we settled on after looking at the criteria was something that's electrochemistry-based. So electrochemistry has been used to prevent corrosion in the ocean, and now we're trying to use the approach to intentionally deliver very low doses of the target nutrients. So in the cartoon over here, you look at two electrodes, one that contains the nutrient of interest and another one just to complete the circuit. It's called a counter electrode. And you have electrical pulses that control how often, how much nutrient is released. So what this approach does is it can be situated near the top of the ocean on the surface where there's sunlight. Nutrient dose can be varied two different ways. One, you can change what's in the electrode, so you know what's the composition of the electrode that controls what can be released. And you can control how you add the electrical pulses. That controls how fast, how often you release the nutrient. And then because we can have very fine electrical control, you can have very low doses that you deliver, and you can deliver in close proximity. You could have many of these electrodes distributed, allowing for low dose local delivery. And these can be powered very easily with wave energy or some sort of marine renewable energy to operate. And the ideal opportunity for these is much more distributed deployment instead of one individual iron deliveries. And when there's no electrical input, the process stops. There's no iron release or nutrient release. So it's very easy to control on-off with the process. And while we've been talking about iron fertilization for carbon removal, we see the process being equally relevant for a lot of onshore aquaculture or phytoplankton growth for other applications that may be happening today. So what I'm going to show you is results from proof of concept studies that were done at PNNL. In order for any of this to become tested in the water for iron fertilization, takes many steps and detailed investigation trying to understand energy and environmental impacts, ecosystem impacts, testing in a closed loop. So some of the opportunity in testing these with the ecological variability could be with coastal aquaculture and seeing how they may impact different species differentially. And then to eventually work our way towards in-water testing. So first and foremost, we wanted to make sure we understood the process without the phytoplankton in the system. So what you're seeing is a beaker with 3 electrodes. The little coil on the left-hand side is a commercial alloy stainless steel. Then we have a counter electrode that's labeled platinum titanium just to draw the current and complete the circuit. And we have current pulses, and the current pulses correlate with iron. And what we're doing is using a commercial electrode as our iron source. We're conducting experiments with no phytoplankton, and the current pulse correlates with how much iron is released. And we're analytically taking samples to measure and quantify the release rate of iron and the total iron in the beaker as a function of the experiment. So we made sure that we could control, this is an example, but we could control extensively how much iron was added. So then we introduced our phytoplankton. Again, we're working with Picochlorum celeri. What you see is multi-cultivators in the photograph over here with and without the electrochemical iron addition. So it's control versus electrochemical. And we have the same coil electrode, and we apply electrical pulses, and we watch the growth rate using our optical technique in the top panel, and then how much biomass in the bottom panel. What we're looking at is iron chloride chemically added with EDTA, electrochemical iron addition with this pulsing method without any EDTA added, and then no control, no iron is our control, which is just the growth medium. So what we see is both iron addition increases biomass growth, which is not a surprise, which is well established. But what's interesting is comparable growth can be achieved with the chemical plus EDTA, iron, chemically added iron with EDTA versus electrochemical addition. And the nice thing is the electrochemical approach gives us a lot more process control. Here just we're giving you an example of how keeping the same pulse height and the same pulse frequency, we can deliver, by just running the system for different amounts of time, we can deliver different total amounts of iron, which has impacts to growth rate and total biomass productivity. So what you can see here is the extensive control it offers, which can be further augmented by changing the electrode composition and such. But what's interesting is when you have higher amounts of iron, the growth rate is faster, so the darkest green curves here. But what we see is the iron use efficiency per ion of iron that we add. Iron use efficiency is better at lower doses of iron. So 163 ppb has higher biomass, which is really interesting and suggests that having better control means we add less iron into these streams, and that could be very valuable. So finally, we wanted to see how scalable is this, how long will these last? So we did some accelerated testing where we had pulsing of the current on and off, and then we imaged the electrode, the coils of these alloys before and after. So it started out nice and smooth as seen in panel C. And then as you pulse for 2 seconds, you see some small pits, panel D. 20 seconds shows you a little bit larger pits. And then if you hold the pulse for 60 seconds, the pits get larger. So we're trying to closely work with corrosion and de-alloying experts so we understand the mechanism, because once we understand the mechanism, we can identify the best alloy composition so we can avoid some of those chromium and nickel that may be present in the stainless steel, which have bioconcentration concerns. So we're working our way slowly to understand the mechanism to eliminate these considerations. Finally, we wanted to see how we would compare in terms of cost and emissions for using chemical versus electrochemical iron addition. Preliminary assessments suggest that electrochemical addition, given that there's no EDTA, actually is better in terms of cost and also GHG, which is very promising. But we see more opportunity to improve performance and lower costs going forward. This is just really proof of concept. So our next steps are really, we showed proof of concept. There's lots of optimization, and we have not done any ecosystem studies. So there's a lot of work to do, but this is one of many continuing efforts. In summary, PNNL has extensive capabilities and expertise and interest in marine carbon dioxide removal. I hope I showed you that we're leveraging our highly interdisciplinary expertise in material science, algae biology, and ocean modeling, and so on, that we're taking these ideas, new ways of delivering iron from bench through very careful analyses and testing to mesocosm, and based on how things work, hopefully into the ocean someday, based on progress. With that, I'm going to hand it back to Darcy. And
Great.
I'd like to thank my collaborators. Yep.
Thank you so much for your presentation, Chinmay. It seems like research and technology are really pioneering a new direction here, but it will take some time to scale up. I want to thank all of our speakers. Those were really informative presentations. And in a moment, we will start the Q&A portion, and I'll be turning it over to Kevin Travis with the California Ocean Science Trust to moderate the Q&A. But first, we're going to do one final poll question. And that is, I think you'll see it on your screen here momentarily. After today's presentation, your impression of ocean iron fertilization as a marine carbon dioxide removal strategy change? And if so, how? Right. Hopefully everyone's had a chance to fill out the poll. We can take a look at the answers there. Okay, so it looks like Majority had no change, with about equal amounts of more concerned and more hopeful. So thank you, everybody. It's always interesting to see these poll results. And now I'd like to pass it off to Kevin for Q&A.
Great. Thank you, Darcy. And would love to welcome our speakers back to the screen. Thank you for those great presentations and that walkthrough and overview of ocean iron fertilization. We've received many questions, some really good questions, and I'd like to just start with, we got a question about establishing test beds for MCDR research. I think, Sarah, you answered it a little bit in the Q&A, but maybe you'd like to pose this question to our other speakers. What impacts do you think establishing MCDR test beds or research zones would have on MCDR research? And do you think it would be positive or negative? I know many of your talks spoke about site selection being an important factor for ocean iron fertilization, but maybe also those broader considerations that were mentioned, the practical or logistical ones for establishing these test beds.
I can go first. I think there's a lot of value to establishing test beds, partly because some of the challenges are with accounting for carbon or ecosystem impacts or nutrient monitoring. So having a test, and some of these are very expensive to install or develop project by project basis. So if there is a test bed that has the capabilities, that would really help monitor and more reliably generate the data that we need to answer some of these questions.
I don't know if Sarah or Paul have any thoughts on that. I know, Sarah, you typed an answer. If there's any other comments on that one. All right, we can move to the next one here. We have a couple questions from the audience about public perception and the role of social science. So could you all please speak to the question about how public perception or normalizing such climate interventions, if the US takes the lead or even conducts all these activities appropriately, other nations may follow suit. So are social scientists working with you all in discussing the development of establishing norms or enforcement and coordination? Maybe also to add the role of social scientists in conducting field experiments going forward.
Yeah, I'll take a stab at this one. I mean, I think that, the level of activity that EXOIS is kind of advocating for at this point is not to move these technologies into the kind of climate intervention level. So we're trying to be very clear that we are doing the experiments that need to be done to assess whether or not this is a responsible and effective approach. And at that point, I think once the information is available, then that passes into the hands of decision makers and stakeholders and social scientists and society at large to make the judgment call about whether or not this is something that we see would be effective or worthwhile doing. At the stage that we're at, we're involved with social scientists and governance experts to kind of understand the landscape, so that we design EMRV strategies and we choose our sites with an appropriate engagement of society and stakeholders, so that level of activity is being done responsibly. But I want to be careful to disambiguate kind of our activities at this like pilot stage versus moving any of this, or the desire even to move any of this to an actual production stage. We're not ready to, we don't advocate for that as a group, and we're not kind of ready to advocate for that without the necessary information.
Yeah, I mean, I would say none of us are social scientists, so we're not. It's not necessarily our field, but I think that everyone kind of working in, or a lot of people working in MCR are kind of very aware of the need to involve a broad swath of people in pilot projects or anything that kind of move forward in this. I kind of mentioned that controversial 2012 experiment off of Haida Gwaii. I think that was, kind of a cautionary example to people of kind of how not to do it, because it did not have a broad kind of input before it happened, and it kind of left a negative effect on a lot of people's perception of the whole process. And so I think that there's, kind of a cautiousness in going forward and making sure that that there's kind of a broad understanding across kind of society as things happen.
Just to briefly add to that, I think, so we've been doing a lot of community engagement around CDR at large, besides MCDR. And what we've learned is communities are interested in the benefits and the impacts. So I think, Paul, you did a really good job of sort of thinking about potentially this could be beneficial to fisheries, and that's something they would like to hear, as well as what else they need to worry about, sort of laying that picture, a complete picture, but also backing up with data. So I think there's interest in sort of seeing some of that information laid out for the communities and the conversations we have had so far.
Great, thank you. I'm going to combine a couple questions here around ocean iron fertilization feasibility. So the question is, are the areas where OIF would be most effective the areas where excess nitrates and other nutrients found? There's another comment here that regulators on the West Coast are considering mandating restrictions on different macronutrient additions from land inputs due to concerns about local ocean acidification or hypoxia enhancements. So how does that kind of potential consideration reconcile with the efforts to maximize the use of available macronutrients offshore, for example, with ocean iron fertilization?
Well, the key point, it's not where the excess nitrogen is, it's where the iron is limiting phytoplankton productivity. So you can go, to coastal regions, they're not typically limited for iron. Iron fertilization won't do anything to enhance productivity in those regions where productivity is already really high, unless iron's limiting. Macronutrient enhancement is certainly a strategy that can be used to increase phytoplankton productivity, and we actually have probably been doing that already, along with our climate engineering by the burning of fossil fuels. We've probably been stimulating phytoplankton productivity. We know already with dead zones in the Gulf of Mexico that we've altered those ecosystems in a similar way, but it's different. It's different from the iron addition and promoting productivity. So those are kind of two different approaches and two different types of ecosystems, I would say.
Thank you for that. And I might go right back to you, Sarah. There was a question regarding SF6 that you wanted to bring to the discussion here. I think it was about using alternative tracers. Maybe just throw it to you for that one.
Thanks. There were a couple questions about this. Yeah, so sulfur hexafluoride is a greenhouse gas itself. It's added in super small amounts, and it's used to trace, and has been used in prior experiments to track the patch as like an important part of figuring out where you added the iron once the iron is gone. It only lasts for maybe 2 weeks at the most. And the concerns that I was seeing come through in the chat are kind of beyond my level of understanding of the use of SF6 as like a natural tracer, like a seawater, to measure seawater. There's an alternative, and I don't even know what the name of it is, but that did come up in some of our field planning discussions. So members of my group are aware that SF6 may not be the right exact chemical form of a tracer to use. We also had conversations about not using an inert chemical tracer at all and just tracking the patch with like a fleet of surface drifters. So I would say that our strategy for how we will actually track an amended patch, if we get to the point of doing these field deployments, is still in development. And I'm grateful for the comments reminding us that we have to be kind of conscious of what other kinds of unintended damage we might be doing by adding something that is a greenhouse gas or maybe interfering with kind of baseline monitoring efforts. So Thank you for those comments.
Yeah, I appreciate those comments and clarifications too on different points that were brought up. Maybe taking a step back, I mean, you talked about the monitoring and tracking of a patch to understand the carbon reduction as well as different unintended impacts. Maybe a question for all of you, maybe starting with Paul, though, about what is needed to develop a more comprehensive and reliable MRV protocol or monitoring, reporting, and verification for ocean iron fertilization for MCDR. So how could maybe an MRV protocol or standards incorporate potential environmental or ecological impacts? And I know, Paul, you talked a lot about this.
Yeah, I mean, I guess it's been sort of described as MRV, focusing on tracking the carbon and whether or not carbon is actually sequestered. And the key kind of questions there have to do with how much of that is actually sequestered in the deep ocean versus how much is just sort of remineralized and brought relatively quickly back up to the surface and the atmosphere. That isn't really my kind of area of focus. My focus is on ecological interaction impacts. And Sarah sort of described that, and it's described other places as EMRV, so the ecological monitoring and evaluation. And I think that's a kind of a key piece of both the development of pilot projects, of trying to understand what's happening ecologically, and in kind of moving forward to any kind of deployment scale activity. And it's a challenging problem because it's happening over a broad scale over long time periods. And so, current discussions have focused a lot on remote technologies and automated technologies to try and do that piece of the puzzle to track what's happening. I don't know, Sarah, if your kind of group has got specific proposals.
Sorry, specific proposals for
EMRV?
Oh, for EMRV, yeah. Yeah, well, we're kind of working on like an EMRV co-design process with social scientists and science communicators and community stakeholders. Like we came in a certain region as natural scientists with an idea of what we think the risks might be. But our perspective on EMRV might be very different than the perspective of like somebody who might be more interested in fisheries. Like from a oceanography point of view, we may just assume that the amount of productivity that we're adding to a certain ecosystem will be so minimal that it'll just be a blip on a normally very kind of high shifting baseline type environment. And we can say, oh, we don't think it's important to measure the impact on fish. That might not be good enough for people who are motivated to understand what the impact on fisheries might actually be. So we're trying to figure out ways of including that kind of feedback into a comprehensive EMRV design process to assess what we as natural scientists believe the risks might be, but also to holistically address the list of risks that anybody who would be aware of this activity and interested in its outcome would benefit from the knowledge. So we're in like the early stages. I wouldn't say that like we have a very well developed list or plan at the moment, proposal, but we're working out like the details of how to do that kind of a process.
Yeah, and I'd say it's a challenging piece because a lot of our kind of bigger risk concerns and the things that we care about are like the effects of nutrient robbing on fisheries. Those are really hard things to test in a small scale pilot. You can't really detect those. Just being able to measure the change in nutrients would be difficult enough. Measuring the change in a fishery that's naturally extremely variable and driven by a lot of other factors, and sort of teasing apart effect of an experiment, extremely challenging. So some of the things that we care about the most are some of the things that are hardest to measure. And we're going to need to rely, I think, a lot on modeling as we try to anticipate what those effects can be, because they're hard to measure directly.
Thank you, Paul and Sarah, for that. I have one quick question for Chinmay on about the technology that you and others at PNNL are developing. What's the current stage of RD&D, and are there similar technologies in the market? And does the current stage mean we can realistically deploy it in the water, given the obstacles we learned about?
That's a very good question. We have seen one other paper where they're using straight iron electrodes to deliver some of the nutrients, but there's not been anything else in the market that we're aware of. And the challenges are, you can't use pure iron because it just corrodes readily. So that control is what we're trying to do with an alloy instead of pure iron. We've worked with commercially available materials with the intention of being able to scale up rapidly. That is the hope, but recognizing all of the research that needs to happen and the permitting and all of that, the one key difference I see is the possibility to do localized low concentration deliveries. So if there's let's say if permitting allows for a minimum, like an envelope or below the envelope delivery of nutrients, then that may be an opportunity that this approach could address that large additions, single addition may not be able to do. But that's to be seen. There's a lot of unknowns at this point. Yeah.
Understand. Thank you. Alrighty, well, we'll have to end it there. We are at the hour. So I just want to say thank you again to each of our three speakers and the contributions and time to this webinar. And thanks to everyone for attending and the engagement. We had some really great questions. A recording of this webinar will be available online on all of the different co-hosts' websites. Please stay tuned for our next announcement in this webinar series coming soon. And we encourage any welcome feedback, suggestions on different topics that we can tackle through this partnership and series. But with that, thank you all and take care.
Thank you.